Hi, my name is Robin Cash, and I'm from the University of Melbourne. And I'm going to present a brief overview of personalised brain stimulation, focusing on a technique known as transcranial magnetic stimulation, or TMS. This personalised approach aims to optimize TMS therapeutic outcomes by aligning brain stimulation targets with person-specific brain network architecture. So TMS is most commonly used therapeutically for treatment-resistant depression, where it is typically targeted to the dorsolateral prefrontal cortex. And over the course of several weeks of daily treatment, some people respond very well, but many do not. And the response rate is around 35 to 50%. This is not bad, considering this is a treatment-resistant population, but nonetheless leaves a lot of room for improvement. One basis for this variation may be clinical or demographic factors, although these have shown somewhat weak and inconsistent effects across studies. Another potential factor is suboptimal spatial targeting. However, over the past 2 decades, the optimal TMS target site has remained elusive. So a range of different targeting methodologies have been explored. And you can see here that these have targeted quite different subregions of the dorsolateral prefrontal cortex. Without going into too much detail though, connectivity-based targeting strategies, and in particular, targeting strategies that are informed by an individual's brain connectivity, show the most robust relation to TMS therapeutic outcomes. So which brain connection should we target? Several studies have shown that TMS treatment outcome relates closely to functional brain connectivity between the dorsolateral prefrontal cortex stimulation site and a deeper brain region known as the subgenual cingulate cortex. This provides an obvious opportunity to improve treatment outcomes by prospectively selecting the DLPFC stimulation target site based on optimal connectivity with the subgenual cingulate cortex. One approach would be to identify a single one-size-fits-all DLPFC target site that should work for all individuals, defined by a group average map of SGC functional connectivity across the spatial extent of the DLPFC. By the way, it's worth adding that the initial reason for focusing on the subgenual cingulate cortex is that this region was consistently implicated in antidepressant response across a range of different therapies. On this basis, the subgenual cingulate cortex was also selected as the target region for deep brain stimulation. A potential disadvantage of a one-size-fits-all targeting approach is that there is substantial inter-individual variation in brain network architecture, particularly within the prefrontal cortex, as highlighted here in hot colors, yellow and orange, corresponding to increased evolutionary development within this brain region. These areas can vary to such an extent that individuals can be reliably identified from a large sample based on their functional connectivity fingerprint. A few studies that delivered TMS to a singular one-size-fits-all group target using neuronavigation haven't really shown any compelling improvement in treatment outcomes relative to conventional targeting. So together, these findings provide a strong rationale for personalization of the DLPFC target site and defining this site according to connectivity with the subgenual cingulate cortex. But in order to translate this approach clinically, we first need to know whether precise personalized targeting is even feasible. If personalized targeting based on brain connectivity is feasible, then it should be possible to reliably identify personalized targets that remain near identical in position across 2 different brain scans. However, when we started this work, a paper had just been published using conventional computational methodology, indicating that targets differed by an average of 3.5 to 4 centimeters between 2 separate scans for a given individual, even when using the best quality brain scans. In other words, personalized targets were not reproducible and personalization did not appear to be feasible. So without going into detail, we developed a computational personalization methodology building on previous work by Fox and colleagues from 2012. This ultimately enabled us to precisely and reliably pinpoint optimal personalized connectivity-based stimulation targets. We verified this in 1,000 individuals from the Human Connectome project, that the sites that we identified varied by only 2 millimeters or a single brain voxel between scans repeated the next day or even 1 year apart. Importantly, these personalized targets were not only robust, but also differed substantially between individuals, demonstrating that our approach preserved inter-individual differences. Interestingly, we also had familial data from the Human Connectome project and found that targets were more similar between monozygotic twins compared to dizygotic twins, non-twin siblings, and unrelated individuals, suggesting that these targets are stable and under genetic control. We then tested whether these targets were clinically meaningful in a sample of 26 individuals who had previously undergone a brain scan and who received TMS for depression and for whom the stimulation target and treatment outcome was known. We retrospectively computed the optimal personalized stimulation target from each scan. We anticipated that individuals whose actual stimulation site was closer by chance to their optimal stimulation site, our target site, would have a better treatment outcome. So here you can see the scatter of optimal personalized stimulation target sites across this cohort. And here you can see that we confirm this hypothesis. This figure shows that individuals who serendipitously received TMS closer to their optimal stimulation target site consistently had better treatment outcomes. Moreover, these findings have since been independently replicated by Shan Siddiqui and colleagues in an independent depression cohort. In parallel, other groups have been working on this, and there is now preliminary prospective evidence emerging that connectivity-based personalized targeting may enhance clinical outcomes across invasive and non-invasive brain stimulation modalities. So across 6 separate but small studies, including an RCT just published in the American Journal of Psychiatry, the average response rate is 87% and average remission rate is around 65%. This is vastly higher than with conventional targeting, where the average response rate is 46% and remission rate is 31%. And these findings are, in my view, some of the most exciting in the field right now and clearly warrant larger clinical trials. The importance of brain connectivity in mediating clinical outcomes is also emerging with another form of brain stimulation known as deep brain stimulation. Several sites around the world are now implementing this methodology, and in Australia, the approach is now TGA approved, so the equivalent of FDA, and has led my colleagues to launch the first Australian clinic to offer personalised targeting. Now, a key question is whether the benefits of personalisation, the benefits of personalised targeting based on brain connectivity would extend to other TMS applications. For instance, previous work has shown that multi-session TMS to the parietal cortex can enhance associative memory performance, the parietal cortex being a key component of the cortical hippocampal memory network. As you might have noticed before, the parietal cortex is another brain region that has high inter-individual variation in brain connectivity. So without going into detail, we retrospectively analyzed an existing data set where MRIs were acquired and this multi-day TMS protocol was delivered. We knew the actual stimulation coordinates and applied our personalization methodology to compute individual specific or person specific optimal target coordinates. Notably, the original coordinates were personalized, but our approach offers some advantages, for example, in terms of the signal to noise ratio of the 20 voxels conventionally used to define the hippocampal seed. These data showed once more that closer proximity to these optimal personalized targets was associated with better TMS-evoked improvements in memory performance. In this slide, I just wanted to make the quick point that TMS tends to impact and modulate distributed brain networks rather than just a single brain region, such as the local stimulation target site. So here you can see that changes in brain connectivity occurring with real, but not sham TMS across different regions in this cognitive network following stimulation of the cerebellum. With no change in the control site. Other research has indicated that the most therapeutically effective TMS and DBS targets, as well as lesions eliciting symptoms, are functionally connected to the same disorder-specific brain circuit. And Shan Siddiqui's work has delineated a convergent brain circuit in depression. This becomes relevant, as I'll show you on this slide. So a recent meta-analysis demonstrated that across the past 20 years of task-based imaging research into emotional and cognitive dysfunction in depression, comprising almost 100 neuroimaging studies, there was no convergence in terms of the anatomical loci that were identified as showing aberrant activity during these tasks. The lack of convergence was proposed to potentially be attributed to different and novel task designs across different studies, as well as small sample sizes and various other factors. In my latest research, I considered whether these regions might be linked or functionally related from a connectomics point of view. I will skip the details of the methods and null conditions, but in short, these divergent coordinates actually localized to statistically highly robust distributed brain networks. Importantly, these networks recapitulate clinically meaningful and independently derived models of depression circuitry. Moreover, when I use these networks as a template to define personalized TMS targets in the same way that I described earlier, TMS therapeutic outcome was dependent on how effectively this emotional dysfunction circuit was targeted. So lastly, many thanks to my mentor, Andrew Zaleski, and to those who shared data with us, including Paul Fitzgerald, Simon Eichhoff, Veronica Müller, Josh Hendricks, James Coxon, Shan Siddiqui, and Mike Fox.